This tutorial will focus on DNA replication. This is the process by which DNA makes an exact copy of itself. DNA makes an exact copy of itself just before the cell is about to divide, in other words, to go through mitosis. So, since mitosis leads to two new cells, two new daughter cells, each cell has to have exactly the same DNA. So DNA has to exactly replicate or copy itself. Before we look at that process in a little bit of detail, I would like to just show you something else you need to be familiar with, which is something called the cell cycle. The cell cycle describes the stages that a typical cell would go through growth, DNA replication, and then division. The cell cycle is summed up in this diagram here. So let's have a look at what it's showing. So essentially, after a cell is formed, which will be around about here, that is after cytokinesis, we might just make a note of that. So cytokinesis is the actual division of the cell itself, keeping in mind that mitosis is the division technically of just the nucleus. So cytokinesis will form two new cells. For each cell, they will then go through a growth phase. Alright, so their normal growth phase, so that the cells grow bigger through this phase here, and this is referred to as the G1 phase. So growth phase 1. The cell is heading towards further division. Then after the growth 1 phase, G1 phase, comes the synthesis phase, or the S phase. During the S phase, DNA makes an exact copy of itself. In other words, it goes through DNA replication. So this is the S phase or synthesis phase. Synthesis means to make. After the DNA is replicated, the cell continues to grow through what's called the G2 phase and prepare for mitosis. So remember, mitosis is the division of the nucleus in a cell. So the cell enters the mitotic phase, or the M phase, and goes through the process of mitosis, which you will have learnt about before. The various stages of mitosis, such as prophase, metaphase, anaphase, and telophase, and then finally cytokinesis again, where the cell will divide into two, and away we go again. So this is what's referred to as the cell cycle, and it's this section here, DNA replication, that we're particularly interested in the rest of this tutorial. So let's have a look at what's involved here. In DNA replication, the DNA molecule has to make an exact copy of itself. This kind of replication is referred to often as semiconservative replication. Semiconservative replication. To conserve something means to keep it as it is. So the term here, semi-conservative, means to keep half of the DNA the same as it is originally, and then to create a brand new strand to add to it. In this way, if you start with two original strands of an original DNA molecule and then add two new strands, then you'll end up with two new DNA molecules. So let's have a look at the process that leads to this. So semi-conservative, remember, refers to the fact that of the two new DNA molecules, one of the strands will have belonged to the original molecule. It's conserved. So, to replicate a strand of DNA, which of course, as you know well by now, is a double helix that's bound up very tightly with histones and highly folded and looped into a chromosome, this DNA will need to be unfolded and then split apart so that a new strand or new nucleotides can be added. So the molecule, the key enzyme molecule that's involved in unwinding the strands is an enzyme called DNA Helicase. And you need to be familiar with this enzyme. You need to know what it is and what it does. So its job is to unwind the double helix so that you end up with two separate strands in the DNA molecule. In the process, there's also some stabilizing proteins involved. These aren't all that important in terms of your knowledge, so just be aware that there are some stabilizing proteins involved in the process. The other key molecule that's involved is another enzyme called DNA polymerase. And there are a couple of different kinds of DNA polymerase. The one that we're interested in here is DNA polymerase 3. So this is an enzyme that's going to add new nucleotides together to make a new strand of DNA. So just like we looked at the use of RNA polymerase when we talked about the making of messenger RNA, DNA polymerase is involved in making a new copy of DNA. Let's have a look at what goes on. I'm going to clear the screen for a moment, clear the ink, and then we'll move down and have a look at what goes on. Alright, so once the helicase has unwound the DNA into two strands, it's important to understand how the nucleotides are added together to make two new strands of the DNA. There's a simple rule that's followed here. The new DNA strand is constructed always from the 5' to the 3' direction. So the new nucleotides, as you can see here, are added from the 5' end of the DNA through to the 3' end. So the direction of addition on this strand goes this way. On the other strand it has to run in the opposite direction because, as you know, the two strands of DNA run in opposite directions. One is 5' to 3', the other one is 3' to 5'. So the two new strands of DNA have to be added in two different directions. On this strand here, up the top, alright, where the 5' is down this end, the new nucleotides can be added in one continuous line. So basically, to start DNA replication, a small segment of RNA is constructed by RNA polymerase and is added as what's called a primer. A primer is something to start something off. It primes it. So an RNA primer is just a few nucleotides long and is added temporarily to the free strand up here. So this is the original DNA strand. The RNA primer is added, and then what the DNA polymerase does, so that's this enzyme here, is it then takes nucleotides and adds them in sequence down the line. Alright, attaching them to the RNA primer and then linking them end to end down the line. Now, which nucleotides go where is determined, of course, by the bases on this side of the DNA. So remember, it has to be complementary base pairing. A's have to be bound to T's and C's to G's and G's to C's and T's to A's. And you already know this. So the nucleotides that are added in order along here to form the new strand have to be complementary to the bases on the original DNA strand over here. So in this way, you build up a new double-stranded molecule, as you can see here. This continues all the way down, and as the helicase continues to unzip the DNA, it'll continue all the way to the end. On the other side of the DNA, this is called the lagging strand, and we'll have a look at that in just a moment. So this is the leading strand, from 5' down this end down to 3' towards this end, and it builds the new strand in one continuous go. Let's now have a look. If you want to pause this for a moment and have another look at where I've drawn it, because I'm going to clear the ink in just a few moments and shift the diagram up. Alright, so that's synthesis on the leading strand. Let's now have a look at synthesis on the lagging strand. This is a little bit more complicated, so let's see how it works. The lagging strand is the strand opposite the leading strand. So we're talking about this strand here. Now remember, the new nucleotides have to be added from the 5' to the 3' direction. And the 5' on this side is from here down to here is the 3'. So on this side, the nucleotides have to be added going this way. Because of this, a single strand cannot be added directly, and it has to be added in small fragments at a time. So what happens is you have a primase, an RNA polymerase that adds a short primer, RNA primer, to start off the process. You've got DNA polymerase 3 here, which will start to add new nucleotides, new DNA nucleotides, that again complement the sequence on this side of the DNA molecule. But these fragments are only added short bits at a time. And you'll see that another RNA primer is added here, and then further DNA is added, and then this happens all the way down. So an RNA primer, I'll change colours for a minute, the RNA primer, along with the short DNA fragment that is added, is referred to as Okazaki fragment, because it was discovered by a scientist, Okazaki, a Japanese scientist. So these fragments, so here's an Okazaki fragment, here's an Okazaki fragment, and so on. So these fragments are constructed section by section, and then DNA polymerase 1, a different enzyme here, comes along and digests or breaks apart the RNA primers and replaces them with DNA nucleotides to fill in the gaps. So DNA polymerase fills in the gaps as we move along the strand. So this is referred to, or this is the lagging strand, and the way it constructs its new double-stranded molecule is slightly different to the leading strand over this side. In the end, though, you end up with two new double-stranded DNA molecules. Let's have a look at the result. So here's a diagram that gives us some sense of what the final result will be. So here's our original DNA molecule, split into two by the DNA helicase at this point, and what you end up with is two new DNA molecules, one here and one here. Alright, keep in mind that one of the strands of the new molecule, so this is one new molecule here, one of the strands, the dark blue, is still the same strand that belonged to the original molecule, and the light blue one here is the new strand. So semi-conservative replication. One strand from the original molecule plus a new strand makes one whole new molecule of DNA. Likewise on the other side, here's the original strand, it's conserved, and here's the new strand, hence another new molecule of DNA. Eventually the entire molecule, of course, will be replicated, and you'll end up with two quite separate molecules of DNA. So from one original strand you've now got two strands, both of them absolutely identical to the original. So this is DNA replication. Again, it would be very helpful to have a look at a YouTube clip. Type in DNA replication into YouTube and have a look at this animated, and it may give you a better sense of exactly what's going on. So here's that idea again of so here's a better picture just summing up that last point of semi-conservative replication, so that one of the strands from each of the original DNA molecules forms part of the new molecule here and here. And these are the two new strands that have been added.